Ladies and gentlemen, today my name is Gawel Solosky from Government University. Today I will talk about lignocellulose for treatment using using uh, uh, white rods and bacteria comparison. How this composition works and efficiencies to produce polyalkyl hydroxylonate, which is material which is supported to be substituent to polyurethane. It can be used from algas and for fermentation rest can be used. But here, Professor Ragaron brought the form the method of degradation using the lignin from wood flour, so the sodas coming, which is a very big waste of furniture, and how it will in comparison to different methods will be digested and compared. This digestion is important because this can give a green method of polymers, and the pine wood was a most useful. And as Professor so, Ragauskas use for this white rod and as the most efficient. But here is the disintegration given in different conditions in thermophilic and mesophilic conditions. So, and using different bacteria to show in different cases how it disintegrates goes. The first was composting method. For composting was mixing of many waste, and the waste was mixed with the sawdust, rabbit food, and vegetable mature compost for flour, sugar, corn, and urea. This was the mixture for checking different cases. The test after this lignocellulose way treatment was tested using characteristic was by thermographiometric analysis, and these results were given in the tables. The studies was using the compost after how was also checked in 10, 21, and 37 days of composition, how it works. The results were given that first was the wood flour was 15% of cellulose, and theirs is the the processing was used and how it was compared also with how is cellulose and how this wood flour is goes. And in case of after treatment, it was using in case of dry wood was in case adding of cellulose was less was more dried about 2%, and then was showing the parameters about how with cellulose and wheat flour was working, and the thermographiometric analysis shows that maximal temperature was 2094 to only cellulose with mixture, and wood flour was 4 degrees lower, and so very similar material. The assessment of biodegradation was due to the days, and was giving that wood flour and this mixture was the same. Was disintegration was the going 45 days, and the most the most rapid was at 25 to 40 to 15 days. In the first 15 days, the degradation was very low in every case. And case of of adding wood flour, not cellulose, the disintegration was slower than in case of adding cellulose to the composting material. So wood flour is not, but is most useful as waste. And still the adding of cellulose only by waste is very much usually lower. But in case of adding cellulose was after in the last case after 45 days was very big in case of adding cellulose, while wood flour give the lowest in every case. The case is showing that how is degradation. This causing the showing how is utilization made, and it is using how in different cases where working fungi and bacteria, fungi and white rot and bacteria thermophilic and mesophilic. In case of the alone only for composting without adding of cellulose or wood flour, the mixture, the fungi population was similar. In case of wood flour, it was big. This grow only 21 days and no further. In case of the cellulose was much different because after 10 days cellulose was grown then decreased and finally was big increase after 30 days. This showing that fungi develop in case of cellulose so much keys. In case of white cells, of white rot fungi, not brown rot as in fungi, the population was during days in case of composting decreasing. In case of adding the compost to a composting material, cellulose was increasing, the highest in the 37. Similarly, in case wood flour, the increase was the increase was higher than in case of cellulose, and because the white rot used this lignin, so better conditions for using. In case of mesophilic bacteria, in case of composting, after some days, after 21 days, the mesophilic bacteria decrease population. In case of wood flour, the increase is wood flour, like in brown rot, is gradually increasing much more than in case of adding cellulose to compost material. In case of thermophilic, the population was much stronger than in mesophilic. So better conditions in composting was for thermophilic than mesophilic bacteria. This shows the differences how it will increase and why this biodegradation is made. This biodegradation allows to, to remove lignin, and this is the important work because it was published in Polymers this year, showing how this lignin is increasing degradation. The the fungi was good at initial stage very well, and cellulose, adding cellulose give back the biggest degradation or disintegration, while food flour is the lowest. So the addition to some cellulose is a very good idea, while some other hard ways, like wood flour, is rather not favored to increase decomposition. The fungi are fine in the decomposition, grow in initial stage, but mesophilic and thermophilic grows after 30 days. That's major factor. So this shows that this integration of biological methods for to lignin, the removal from lignosus material, can be combined with bacteria and fungi, because on different time would be firstly by fungi, and then after material is unfavored to decompose so well by fungi, the bacteria are coming more willing because are simpler and much less energy is required. So thank you very much for listening me.